Welcome back to Just Biotech Biotechnology Tutorials. Today we are going to study about the purification of DNA from living cells. That is how we can separate out our DNA for our genetic gene cloning and DNA cloning studies. How we can get the DNA complement out of the cells, and we can prepare the solution of DNA from different kinds of cell like bacterial cell, plant cell or animal cell for a further gene cloning studies. So to start with, let us consider what kind of DNA are required for any particular genetic engineer for the gene cloning experiments. So the DNA that can be required can be of three types, right? It can be total cell DNA, or like it consists of total cell DNA. What it consists of? It consists of the genomic DNA as well as the additional DNA whatever is present inside the cell. So it may be plasmid DNA or other DNA component present in the cell. So total cell DNA consists of the genomic DNA, or you can say chromosomal DNA, as well as the other DNA forms. Now the next type needed by genetic engineers is pure plasmid DNA. When this kind of plasmid DNA is required, when we are using the plasmid as a gene cloning vector, right? As I described in my earlier videos about the vectors. So if we are using a plasmid vector, you will require a pure plasmid DNA solution. So for that we need a plasmid DNA solution. Why do we need cell DNA or the chromosomal DNA? Because if we require the genes for cloning or for studies, so this will help us, or this will provide us as the source of those genes that are required to be cloned. Now next, if we are using a phage particle or phage DNA as a vector in gene cloning experiment, we will require a phage DNA solution. So this total cell DNA can be any DNA complement from bacterial cell, plant cell or animal cell. Like plasmid DNA, generally we get it from bacterial cells. So the steps for the total cell DNA and the plasmid DNA are almost similar, but somewhere on the later stages we need to separate the plasmid DNA from the total cell complement of the DNA, right? Or the chromosomal DNA. That is the basic difference between the process of total cell DNA and plasmid DNA isolation. The phage DNA are mostly, recovered from phage particles and not from infected bacterial cells. So that is like the isolation or the separation of chromosomal DNA is not a problem, usually not a problem, right? So next, let us first see the basic steps of total cell DNA preparation. What are the basic 4 steps of total cell DNA preparation? In this video, I'll be talking about total cell DNA preparation only. The plasmid and phage DNA preparation will be tackled in next video. So what is the first step? See, this is our bacterial culture. We have the bacterial cells growing up here. What is the first step? A culture of bacteria is grown and then harvested. First of all, we will inoculate our bacteria into a culture where it can grow up to a certain amount or number of cells, and then we harvest or we separate the cells, or the bacterial cells are harvested from that culture. Now, what is the next step? We actually extract out the contents of the cell. That is, we obtain a cell extract. How this happens? We achieve cell lysis, or we break out the cells so that the contents of the cells are released. So the cells are removed and broken to give a cell extract. This solution, where we have the contents of the cell and the cells are broken down, is known as cell extract. Now, what is the next step? What we do is, from this cell extract, which contains DNA, RNA or protein and the other cell debris, we have to purify our DNA. So we have to prepare a pure DNA solution from this. So the third step is to purify the DNA from cell extract. Now once we have the pure DNA solution, the next step is to concentrate our DNA for our gene cloning purposes. Usually for during purification we actually go through procedures which dilute our DNA solutions. So for the gene cloning purposes we have to concentrate this DNA solution. So the last step is concentration of DNA solution, right? Now, let us first consider the very first step of this DNA preparation, total cell DNA preparation, that is the growing and the harvesting of a bacterial culture. What happens with the bacterial cells? The most easiest ways to grow them is in the liquid cultures. They grow very efficiently and effectively in liquid cultures. So, what is the purpose of the liquid culture? The medium or the culture medium that we use, it should provide essential nutrients and elements that will allow the bacteria to grow effectively and divide to give rise to a ample amount of population, bacterial population. So there can be two types of media that we can use here. One can be defined media and other can be undefined media. What is a defined media? If the exact composition of the culture media, in terms of its elements, minerals is known, it is known as defined media. So example of a defined media is M9 medium. It is an inorganic medium which contains the elements like nitrogen, magnesium, calcium, and the energy sources like glucose. So what is the limitation with defined media? With this media, we actually need to, add a few supplements before we can actually achieve bacterial growth. So supplements like vitamins and other growth factors are added into this medium to achieve proper bacterial growth. And the next type is undefined media. Because of few components of this media whose exact composition is not known, the exact composition of this particular media is unknown, so this is undefined, right? Example is LB medium or Luria Botany medium. So what does it contain? It contains tryptophan and yeast extract. The tryptophan serves as the amino acid source. Yeast extract will give you nitrogen complement. So the exact composition is not known for tryptophan and yeast extract. That is why it is known as undefined media. But the main advantage of using an undefined media such as LP media is that we need not add any further supplements, that is growth factor or vitamins, to achieve bacterial growth. We can just use this undefined media. So what happens if we grow bacterial cells in LV media at a particular conditions like 37 degrees Celsius, shaking it at 150 to 250 rpm in a rotatory shaker or such, that the E. coli cells will divide once every 20 minutes, that is the usual cell cycle time, and the culture will reach at a maximum density of about 2 to 3 into 10 to the power 9 cells per ml. So that is the rate with which E. coli cells will divide. This growth of the culture that we are culturing, it can be monitored by using spectrophotometer, that is within and it is done at usually at 600 nanometers. So the growth of the culture can be monitored by reading the optical density at 600 nanometer, and where which like the 1 OD will correspond to 0.8 to 10 to the power 9 cells per ml. That is the way of monitoring the cells, like the how much cells do we have on the culture. So we use the spectrophotometry in this technique. The next step is the harvesting. Now, once we have achieved a certain amount or the certain number of cells in the culture, we can, measure it by using spectrophotometer. And once we have an ample amount of cells, the next step is to separate out those cells from the culture media. How do we do that? We do the harvesting of the cells by using centrifugation. when we centrifuge the culture medium, the pellet which we receive contains the cell. So this harvesting of the cells is done by pelleting the cells in a centrifuge and pouring out the culture media. The supernatant remains in the the culture media remains in the supernatant, which can be poured out, and the cells can be recovered from the pellet and can be resuspended in the smaller amount, let's say 10 ml of the solvent. So that is all about growing and harvesting a bacterial culture. Now what we have the grown bacterial cells in the pellet which we have resuspended. So we have ample amount of number of cells from which we can isolate our total DNA. The next step is the preparation of cell extract. What is cell extract? After breaking the cell, release the components are released into the solution. The components of the cells are released, and that gives you cell extract. So as we all know, the bacterial cell is surrounded by a plasma membrane, which is itself surrounded by a rigid cell wall. So the techniques for lysing the cells will involve breaking of the rigid cell wall as well as the plasma membrane, and then only the contents of the cell will be released. So there can be two types of methods, maybe physical methods where we will use the mechanical forces, and the next step is the chemical methods. With the chemical methods, we will use the chemical agents, and this is the method which is preferably used with the bacterial cells when our objective is DNA preparation. So in the chemical method, we will use two or three reagents. The first reagent, it will attack the cell wall, and the second reagent will attack on the cell membrane. So let's say with E. coli, the cell wall, first we do the weak attack the cell wall of the E. coli by using the enzyme lysozyme. From where do we get this lysozyme? It is present in the secretions like tears, saliva, and other stuff, and it's also present in egg white. So this enzyme, it digests the polymeric compounds which are present in the cell wall and are responsible for the integrity of the cell wall, right? So first we weaken the cell wall by lysozyme, and most commonly it's not used alone. It is used with EDTA or ethylene diamine tetracetate. It is a chelating agent. So why it is used? It will chelate out, or it will remove the magnesium ions, which the presence of magnesium ions is very important for preserving the overall structure of the cell wall or the cell envelope. So EDTA, you can say it is easing, or it is helping lysozyme to break the cell wall. And most commonly a detergent such as SDS or sodium dodecyl sulfate, it is also added along with EDTA and lysozyme, which cause the lysis of the cell membrane by removing the lipid molecules or by breaking the lipid molecules. So in the chemical disruption of the cells, what we are using? EDTA, lysozyme, and SDS. So after the cells are lysed, the insoluble cell debris is also removed as a pellet, and a clear cell extract is obtained in supernatant. So after the lysis of the cells, we will carry forward with the centrifugation step, and by centrifugation the cell debris will also be removed, and that is how we will obtain the clear cell extract in the supernatant, right, with the actual centrifugation and the obvious centrifugation technique, right? So now we have the cell extract, we have broken the cells, we have released its components, we have harvested and cultured the cells, removed the cells as a pellet, broken them up, and now the next step is the like uh, next step would be purification of DNA from cell extract. So this is what happens during the cell preparation of cell extract. This is cell lysis. We disrupt the cell wall and we release the contents of the cell, and then we use the centrifugation method to remove the cell debris, and we get the cell extract containing DNA, RNA and protein in the supernatant, right? So let us purify our DNA from the cell extract now. The cell extract, it contains DNA, RNA and protein, right? So there can be actually two methods by which we can purify our DNA from this cell extract. One, one approach can be degrading all the other contaminants. That is, we can degrade RNA and protein and we can then get the pure DNA. The next approach can be we can just separate our DNA from protein and RNA. So one is degrading the contaminants, the other is separating the contaminants. So for these two approaches, we have organic extraction and enzyme digestion. This will involve the degradation of the contaminants, and the next approach is ion exchange chromatography, wherein we will separate our DNA from the contaminants. So let us first look into the first approach, that is organic extraction and enzyme degradation. This will involve the degradation of the contaminants. So the standard way to deproteinize a cell extract is to add phenol or a 1:1 mixture of phenol and chloroform. What happens if we add phenol and chloroform into a specific ratio, that is 1:1 ratio, to the cell extract? What will happen? It will precipitate the proteins along with it, right? So let us suppose this is the cell extract and we are adding phenol and chloroform and we are mixing with it. What will happen? The proteins will coagulate as a thick layer at the interface. When we separate the aqueous and the solvent layer through centrifugation, this will see, this is the cell extract. We mixed it with phenol and the chloroform mixture. Then we centrifuged it. What will happen? The phenol or the solvent layer will be at the downside. The aqueous layer containing DNA and RNA will be in the supernatant, and the proteins will be precipitated or coagulated at the interface. So what we can do? We can simply pour out this aqueous layer containing DNA and RNA, right? So this is how we have extracted DNA and RNA and we have degraded or precipitated proteins, right? What happens if we have very high concentration of proteins in the cell extract? One thing we can do is we can repeat this phenol extraction once again. So we can have a cycle of phenol extraction, but what is the disadvantage? It will result into the breakage of DNA at like in each cycle. Some breakage to DNA will be caused. So for that situations, there are proteins like pronase or proteinase K, so which we add to the cell extract before the phenol extraction, and that will breaks the polypeptides, so that helps in phenol extraction for us. From this DNA and RNA, we can actually remove the remaining RNA by using the enzyme ribonuclease, which breaks down RNA. So in the end we will get a pure solution of DNA, right? The next approach is ion exchange chromatography. What is the principle of ion exchange chromatography? It separates molecules according to how tightly they bind to electrically charged particles present in chromatographic matrix or resin. So let us say we have the chromatography column. We have a matrix placed here and we pass our cell extract from here. So what will happen? All the electrically charged particles, let us say DNA, RNA or protein, will get bound to the resin or the chromatographic matrix present here, right? See here, this is the resin and these are the DNA particles which are getting bound to the resin. So when we are passing the cell extract, what will happen? The salt, what is the function of salt? If later on we will add salt, this causes the disintegration of the DNA or the charged particle from the resin, right? So if we add the cell extract on the top, and if we add gradually increase the salt concentration, if we first add a lower concentration of salt, what we will get in the elute, we will get protein and RNA. Then later on, if we increase the salt concentration, we get the DNA in the elute, right? Why we are getting DNA at the later stage? Because it is more tightly bound to the resin. So if it is more tightly bound, we need a higher salt concentration. So thus we can separate out different molecules one after the another by increasing the salt concentration. So this is how we can purify DNA with ion exchange chromatography, right? The function of salt here is to destabilize the interaction. The next step is concentration of DNA samples. Whatever DNA sample we have got from organic separation and ion exchange chromatography, the next step would be the concentration of DNA sample. How do we achieve the concentration? We do it with ethanol precipitation. So generally with organic extraction, we do not need this concentration of DNA sample, or we do not need to do this step of concentration. Why? Because we usually get a thick, thick solution. So this is a thick solution which we have got through organic extraction. If we will add like in the presence of monovalent cations such as sodium at the temperature of minus 20 degrees Celsius, if we will add ethanol into this thick solution, it will the DNA can be popped out with the help of glass rods and stick to the glass rod, and it will get precipitated, and this long fiber can then be usually be redissolved into small amount of solution. Now if we have an aqueous solution which we have got with the ion exchange chromatography of the pure DNA sample, we can simply add the ethanol and we can separate out our DNA by centrifugation. So this is ethanol precipitation. The basic funda here is in the presence of ethanol and monovalent cations, the DNA gets precipitated out, and later on we can actually resuspend this precipitated DNA into smaller volumes. So that is how we achieve the concentration of DNA samples. So as we discussed about the purification of DNA from the bacterial cells, at some stages in gene cloning experiments, we would need to extract the DNA from plant cells as well as the animal cells. The problem with the plant cell is that the initial stages in the cell lysis, we use lysozyme. So lysozyme, it has no effect on plant cell, right? That enzyme is ineffective for plant cell. So for the plant cells, what we usually do, we grind the tissue in a pestle and mortar. So that is an effective, condition for the plant cells. With the animal cells, there is no cell wall, so we just have to disrupt the cell membrane. That can be usually be done with a detergent like SDS. Okay, so moreover with plant cells, there is one more problem. The plant cell will usually contains another molecule except the DNA, RNA, and protein. It will also contain carbohydrates, right? And the carbohydrates, they are not removed by phenol extraction. So purification is not possible with phenol extraction methods. So for that, we have an advantage of another chemical known as CTAB, or cetyl trimethyl ammonium bromide. So that is the method which we are using here. What happens? What is the property of the CTAB? It precipitates or it forms complex with nucleic acid. So suppose here is a cell extract and we are adding CTAB here. What will happen? It will form complex with nucleic acids, that is DNA and RNA. So after adding CTAB, if we will just give a centrifuge to this cell extract, our DNA and RNA will be precipitated out. So we can just discard the supernatant and we can resuspend this nucleic acid and CTAP complex, and later on the RNA from this can be removed with the help of ribonuclease. So this is a simpler method for isolating DNA from plant cell. There is one more method with where we use guanidine thiocyanate, and it has two properties. The first properties of guanidine thiocyanate is that it will denature and dissolve all biochemical chemicals except nucleic acid. So CTAB, what it did, it just precipitated nucleic acids, and what is done by guanidine, it actually degrades other biochemicals but not DNA. There is one more property of guanidine thiocyanate, that is, in the presence of GT or guanidine thiocyanate, the DNA will bind very tightly to silica particle. So this is what happening. This is a silica particle, this is DNA, and if the guanidine thiocyanate is present, they will bind very tightly together. So like earlier ion exchange chromatography, this is the chromatography column containing the silica particles. If we are adding the cell extract along with guanidine, what will happen? It will denature all the biochemicals, that is RNA, protein, carbohydrates present, and so that can be eluted out or and discarded, and later on if we add water, we can actually recover DNA. So with the help of guanidine thiocyanate, the recovering and the purification is made very easy, right? So this is all about the purification of DNA, of the total cell DNA. In the next presentation, we will study about the purification of plasmid DNA and Phage DNA complement. And just do let me know if you need any help. If you're stuck somewhere, let me know in the comment section, and press the like button, share my video, and don't forget to subscribe my channel, right? Bye bye.